Guys, welcome back. Today we are going to talk about cell reproduction, which is another Stirninger process pointing at this R. Let's go ahead and start. Why do cells divide? Okay, first they have to adjust their surface area to volume ratios. Small cells are more efficient at the transfer of material. Second, all right, they need to grow. We need to add more cells to get bigger. Also, cells get damaged, okay? So we have to repair and replace those damaged cells. This is occurring in all somatic body cells. Somatic means, we'll compare that with gametic, but somatic cells. Cell division in different types of cells. So in prokaryotic cells, there's no nucleus, all right? So they can do some very different stuff than eukaryotic cells. They have a nucleus. That nucleus contains chromosomes, and those chromosomes have to be duplicated before the cell can divide. So eukaryotic cells have to undergo a process called mitosis, which is the process of nuclear division. So what's the cell cycle? The most of the cell cycle is the interphase. All right, this is the normal life of the cell. This is in G1 and G2. That G stands for growing. In growing 1 and growing 2, the cell is simply growing and getting bigger. All right, during the S phase, which stands for synthesis, the DNA is copied. All right, and here we have a lovely image of a doubled chromosome. Mitosis is just a small part of that, and during mitosis, all right, we have a parent cell, all right, that divides to make two identical copies or clones of the parent cell. So the number of chromosomes in the parent cell for a human is 46. And when it splits, we have two daughter cells that each have 46 chromosomes. The process of mitosis takes place in a couple of different stages. First, we have prophase. That's when we start to dissolve the nuclear membrane, all right, and those chromatids come together to form the chromosomes. Then we have prophase, metaphase, all right, as it moves from that prophase into the metaphase. And metaphase, meta meaning middle, all right, the chromosomes line up along the middle. Anaphase, they're pulled to each end. And telophase is where we start forming a new cell membrane, all right, where cytokinesis is the final division where we finally get two independent cells. What happens when mitosis goes wrong? Well, when mitosis goes wrong, it turns into something that you are probably familiar, cancer, because cancer is caused by uncontrolled cell division. All right. And there's various different things that do this activity. First, we have binary fission, like we see here with this paramecium. Then we have budding, which is often seen on coral. Sporulation, all right, which we see on like dandelions. Vegetative propagation, here we see it on this spider plant. And then regeneration, we see this on starfish when we like chop off a limb and that grows a whole new starfish. That's asexual reproduction. Let's talk about sex, baby. Let's talk about you and me. All right, now, no, let's get serious. So with sexual reproduction, there are two parents, you and me, all right? The offspring are different, and this happens by the process of meiosis. Meiosis produces gametes, and we know gametes as the sperm and the egg. All right, so why would we do meiosis? Meiosis results in a lot of different things. So first we have an egg that is haploid, meaning it has half the normal number of chromosomes, and sperm, which is also haploid, all right, having half. And when they come together, we get a zygote that's diploid, di meaning two of each type of chromosome, all right? So there's a lot of pros to meiosis. All right, some cons is it's not as fast as asexual reproduction. You can't produce as many offspring, and you have to find a mate to do it. However, the huge pro that really helps is it results in genetic variation, all right? And that genetic variation is really important to keeping people survivable over time. Genetic variation in meiosis. What happens is first, even before meiosis occurs, we have what's called crossing over. And those are when homologous, homo meaning the same chromosomes, switch pieces to create these different pieces. That creates in lots of different daughter nuclei or lots of different potential eggs or lots of different potential sperm. And this is called genetic recombination. An assortment, okay? Homologous pairs of chromosomes are sorted differently each time, all right? There's so many different ways we can sort these. So if we look down here at this, each of these have a capital T and a lowercase t, and a capital S and a lowercase s. How are the many different ways we can sort them?